I think we'll just get going, okay? Let others join when they come. So, good afternoon. And we were discussing about epigenetic mechanisms. We discussed about it. We learned a lot about the molecular mechanisms of epigenetics, mostly that of DNA methylation. We also learned about histone modifications. And then we also learned about X chromosome inactivation. And we also discussed about several other things about global methylation status of the genome and so on. What we are going to do now is, while this epigenetic mechanisms is the center for the regulation of genes from this set to from one cell differentiating into another cell, we should now also use some technology and an application about how to modify these epigenetic mechanisms. And that is what we are actually going to discuss today. So, what we will discuss, of course, is we are not going to go into details about all these things, but I will still discuss a few of those that will make sense for you. So let's just take a step back and see where we are, and then we will try to know what we should do. So here is a patient. Assume that patient needs some cells which may there was a mutation in this person's genome, so because of which the person is unable to produce a functional organ or tissue, okay, because of which the person is having problems. Or it could also be that we want to test some drugs on his cells or his or her cells, patient cells. Let's just say in either case we need to reprogram. That is our goal. So we would have isolated or taken a skin biopsy, into which we will, using several mechanisms which we will get to it about these factors, we will discuss a little bit, and based on that we could generate induced pluripotent stem cells. And from that induced pluripotent stem cells, we could make a repair by using recombination. So if there is a non-functional gene, we could have repaired the gene when it is in the state of induced pluripotent stem cells, and then again differentiate whatever cell type we want, and then those cells can be given to this person. So here it is, we will call it as de-differentiation, right? Because a differentiated cell is becoming something similar to that of ICM or embryonic stem cell, similar. Because they are induced artificially, we will call them as induced pluripotent stem cells. There is also what we can also do is we can in vitro, we can differentiate them into a cell type, and then we can test different drugs, which drugs are for toxicity or efficacy. And then we can then use whichever drug is working, those drugs can be used in the person to treat if there is an ailment or so. So essentially, what all reprogramming is mostly it is about epigenetic modification, right? Majority of them do come under epigenetic modifications. So this is mesenchymal epithelial transition and also senescence, cells getting old, and so on. So a somatic cell and a pluripotent stem cell, all of them they have their own genetic program based on the activities and mechanisms involved. This is Wnt signaling, which we will discuss later. And in one way, the nuclear program decides the fate of the cell. And if we want to derive a different type of cell, we are supposed to reprogram them. change the epigenome of it. Thereby, the cell type that we are desiring, those genes that are specific to the cell type we desire are expressed, and the others are repressed, so that the cell changes its form and function to the desired type. So here is a paper. I mean, this is a paper, Induction of Pluripotent Stem Cells from Adult Human Fibroblasts by Defined Factors. It's a nice paper. You should also see, this is one of from the lab of Yamanaka in Japan. And the person has won Nobel Prize in 2012. And this is a good paper for several. There are many other associated papers, of course. So this work has done something. This, and there is one more paper. I can't remember the exact details, but what they did was they studied all the literature and figured out, I mean, tried out what the reports. So different labs, lab 1 says X, Y, and Z are important for pluripotency from their results by using different methods or different model systems and so on. Another lab might have said, no, A, B, C, and Z are important for pluripotent factors. Lab 3 may say P, Q, Y, B, C are important for pluripotency. That means these are transcriptional factors that can induce pluripotency. They can make one differentiated cell can be transformed into a pluripotent stem cell. So by increasing these protein factors. So like that, they have read about all the literature, and what they have compiled is they have made that about 20 different proteins or genes are implicated in maintenance of pluripotency. When you say implicated, different labs, there will be differences based on different types of labs, their conditions, many things play a role, and so they have found different labs have different things. So there are 20 genes, and it could be any combination of them that are most essential for pluripotency. So they have thought about a good plan, and what they did was they cloned all the 20 genes into vectors and tried to check if I will say, somatic cell differentiated into iPSC or not. Okay, this is what they checked for. They tried out all the 20 genes, and what they could see is they have pluripotency, induction of formation of iPSC was possible. So now what they did was they took all the 20 genes minus X, and they checked if pluripotency cells are coming up or not, and they found that yes, it is forming. 20 minus A, it is not forming iPSCs. I mean, when you say 20 minus A, all the factors are there except X. In this case, except A, all the remaining 19 were given. Like that, they worked out for 19 combinations or so. And what they found was, I'm just giving you the idea or the logic. It's a fantastic way of doing stuff. Okay. So what do you understand if there is a tick mark here? First reaction, if you see 20 genes you have overexpressed in a cell, and that somatic cell has become pluripotent stem cells. That means these 20 contain all the required genes for induction of pluripotency to happen. In second case, they have used all 19, in which they have just they eliminated X, and what they saw was pluripotency was happening. So what does that mean to you? Is X essential for induction of pluripotency or not? Anybody? Yes? Okay. In this case, the even though X is not present, still pluripotency is induced, which means X is not essential because pluripotency is happening even though X is not present. Let's look at the third one, 20 minus A. So they have given 19 and eliminated A, factor A, and there was no, there is no formation of, there is no induction of pluripotency. What does that mean? It means A is essential for pluripotency. Similarly, C is also an essential because if you're not having C, there is no pluripotency, although the remaining 20 are present. So C is essential. And P, even though P is eliminated, pluripotency is happening, so P is not essential. That is how they worked out to find the combination of genes required for pluripotency to happen. And that's a very smart way of doing things. What they found was that there are 4 different transcriptional factors: OCT4, SOX2, KLF4, and CMYC. We are not really going into the depths of what they do and what all they do. That is beyond the scope of the course at this time. But what you should try to remember is that they are transcriptional regulators or epigenetic modifiers which play a big role in several of the genes. Even in cancers, they might be implicated. So as you're seeing, OCT4, SOX2, KLF4, and CMYC, those are the essential factors for induction, or minimal factors, essential minimal factors that can induce pluripotency. And each of them, they have several genes that they control. They are regulators of several other genes involved in key mechanisms of the cell. If you see a simple network as shown in B, OCT4 controls several of these genes, which each of them signaling pathways. They all have their diverse functions again. And you can also see that OCT4 and SOX2, they regulate OCT4 regulates its own expression, so does KLF and SOX2. And there is a feedback mechanism during transcription. And they're also regulating each other. Okay. And then you find there is a integrated network how to maintain the cell, the pluripotency of embryonic stem cells or induced pluripotent stem cells. So the induced pluripotent stem cells have several functions, and they are involved in all of the different cell processes and mechanisms. Okay. So what all I want you to remember at this point is OKSM, OCT4, KLF4, SOX2, and CMYC. So what we are doing now is we want to induce transcriptional factors. We also call them these as Yamanaka factors after the corresponding author or the lab in which these were characterized better. So we are going to use these factors and transcription will happen. Reprogramming to occur. So SOX2, these factors, OKSM factors, they work on target genes. The target genes could be other transcriptional factors or epigenetic modifiers, and which can also eventually there will be a lot of epigenetic modifiers that are influenced, which will then induce the transcription, the pluripotency. That is in the crux of reprogramming. Please note that there are many other things that are involved, and we have to, if we have to read all the biology, it becomes too huge at this time. But you may also want to remember that there are several other factors, nanog, for example, and there are several other factors, people, different, there are little bit deviances from OKSM. I think if you look at the current literature, people think maybe it's not these four, or maybe the other genes are also involved, and so on. So while we wait for more research and more affirmation or confirmation from other labs, independent confirmation, we can keep it as okay, some are involved, and there is a possibility that some other factors are also involved in it. Okay. So the objective for us is to generate induced pluripotent stem cells, and and that is where we are trying to find out the best ways. Our goal is we want a somatic cell. The somatic cell should become pluripotent. This is a somatic cell or differentiated cell. And the genome, the cells are in different form and function because of their proteome. Okay, right here. Because of their proteome and transcriptome. Transcriptome is also related to proteome. So there are differences between the somatic cell and pluripotent stem cell here. There are differences in transcriptome and proteome. Proteome means it is the whole sum of all the proteins expressed in that particular cell at that time in those given conditions. Transcriptome is about all the mRNA or all the RNA, including regulatory small RNAs and so on, that are expressed in that situation. So it is obvious that if the proteome, if the cells are looking different and having different properties, then it means that their protein and RNA content could be different. Therefore, their transcriptome and proteome are different. What is similar in both of them is the genome, right? The DNA content or the sequence is not going to be changed. So what is actually another difference between these two would be, you call it as epigenome. These are just names. What do you mean by the cumulative of all genetic modifications in somatic cell is epigenome for that. And here is other epigenome. So we want change in the epigenome. We want thereby, or eventually we will also have change in the proteome and transcriptome. But we do not want any change in genome. Why don't we want any change in genome or genetic manipulation? It is if we manipulate or make any modifications to the genome, there is always risk of damage, and there is always risk of cancer formation. Okay, so people keep trying to do, there are two ways of doing. One is to use genetic methods, and the other one is to use non-genetic methods. Okay, we will have a look at it. I will take this slide at a later time. So this is essentially, this is what we are trying to do. In genetic methods, that means you're employing, when you say genetic method, you're employing genes or gene manipulation. Like you could have taken gene for OCT4, you can take gene for KLF4, SOX2, and CMYC, and then put them into a cell, into the somatic cell, for example, and then overexpress them, because of which they would become induced pluripotent stem cells. The goal is eventually to have high concentration of OKSM such that they can travel into the nucleus and act on the genome, so that the cell will change its epigenome and eventually become de-differentiated into induced pluripotent stem cells. When we do this, the risk, there are several strategies people do it. all transcription happens in eukaryotes in the nucleus. So anything we put in, uh, anyway, the point is we need to have the for for dedifferentiation or induction of pluripotency, we need to have high concentration of these proteins in the genome, in the cell. Okay, cytoplasm, they'll be produced and then they'll be carried to the nucleus where they will perform the action. So should we give only proteins? Can we give proteins directly into the cell? What if can we what if we can give mRNA instead of proteins? Or what if we can give genes directly into the cell where they would be transcribed, translated, and then the OKSM proteins are produced? So if you look at this, the best way is to use protein or mRNA, because when we use genes, and if the gene gets integrated into the genome, then the risk of cancers is high. So it is not very useful. So there are several ways of doing this. Gene transduction, where we are putting in genes and then we are reprogramming. You can use viral vectors to do that. This is beyond the scope of your course, but we will have a brief discussion on those, what can happen. So we can use the genetic methods with integration, which may be put onto the chromosome. OKSM factors might be there, they will express, and then pluripotency will come. It's very efficient when we use genetic methods like this, but it is very unsafe because there is always a risk of cancer. There are other ways of doing it. Some of them are non-integrating ways, where the DNA doesn't get integrated. You could put it as an episome in the nucleus. It exists as separately and it doesn't integrate into the chromosome. So these are safer than the methods that we are describing here, but still we cannot say what is going to happen eventually. The recombination can always happen, and we cannot be sure that no change in the genome has happened. There are other ways, as I was describing. You can try to produce the protein outside artificially, all the proteins, and then somehow be able to deliver them into the cell. That is a possibility, but you cannot do it directly because we have seen, because it's technically very difficult. First thing, what you should realize is we have seen manipulation of eggs. We have seen manipulation of blastocysts. All of them firstly, they are large and they have a coating around them, and that is called as zona pellucida, which is a tough um, cover. So it can be used to hold, that will allow, using the suction pipette, we can hold these eggs or blastocysts. They are huge compared to other cells. If we take any of the animal cells do not have any cell walls. Okay, the cell walls are not present, and because of which if we make any kind of punctures, the cell usually is going to die out. The damage will be huge. Whereas when you do it to the egg, this zona pellucida still has a protective, acts as a protection to an extent, at least to maintain its shape and keep the contents inside. That's not the same is the case with the normal cells, and they are actually very small. If they are very small, it's very difficult to make any punctures. So that is one of the problems with using proteins. And how many proteins can we do by microinjection, even if it was possible, is very difficult, right? Then instead of giving proteins, why not give mRNA? instead of proteins, give the mRNA of them into the cytoplasm, where in the cytoplasm the ribosomes will bind and produce more of OKSM proteins. Then we may have sufficient concentrations and then localization into the nucleus and then reprogramming. That is certainly a viable one, but we should be able to somehow deliver the mRNA into the cell. That's one of the problems. And then there are other ways of doing, which are not necessarily using OKSM, but other ways. That is what we are going to discuss today. So there are nuclear transfer, that is somatic cell nuclear transfer. This is good. It gives lot of, you can take cell from patient, right? And then we can develop ICM and then eventually embryonic stem cells from the nuclear transfer cells. That is a good or very efficient or very most natural way of among the comparison, most natural way of obtaining embryonic stem cells that are specific to the patient. But as we have discussed, SCNT is not a very efficient method in the sense we have the percent of increase success rate is between somewhere below 1% or so. And then the supply of eggs is a problem. Then there are ethical issues because people will be offended that we are using human embryos and human eggs and so on. There are other methods where people think they take the cells from the patient and they fuse with other cells using Sendai virus or other chemical agents so that they mix up, and then eventually the cells will fuse and eventually they may produce some cells, okay, some cells with patient specific genes, okay, and also pluripotency. Pluripotency comes from these cells, the embryonic stem cells that we have here, and then the patient-specific genes may come from the patient because the patient is the donor of the cells here. But then the fusion rate is very low, and there is always a risk of this is diploid and the cells are diploid, and then we have 4N, which may occasionally can produce diploid cells, but then there is always the risk of tetraploid or aneuploidies associated with that. And some other people we have already discussed this, right? Cell extract. You can use cell extract from something like embryonic stem cells, and then reprogramming may happen. And then we can also try to use cell explantation, and where we study the we'll say explantation is putting into an organism in germline layers, and then they may transform. There are different reports on that, but what we are going to discuss are some of the methods. It's also part of syllabus. So some of them are protein transduction domains. What is it is somehow, as we were discussing in the previous slide, we have proteins of OKSM. And what about sending them inside into the cell? And we cannot directly do that. Microinjection may not be possible. Then what else do we have? Some of the viral proteins are present, which are referred to as protein transduction domains. That means they are proteins and they can move into the cell. As we will see in the next slide, they can cross over the cell membrane, or sometimes they may form the endosomes, and then the cargo can be delivered there. So usually when you say domain, a protein may be together continuous single polypeptide. This could be a domain, and this could be a domain. The domain that we are talking about here, protein transduction domain, is responsible for the entry of the whole protein into the cell. Okay, so the idea is, what if we used protein transduction domains and make translational fusions. That means this is the promoter. I will say it as OCT4 gene is put here, and this would be the start codon. And here it would have been a stop codon, but we are removing it and adding a protein transduction domain in continuous reading frame and then a stop codon. That is called a translational fusion. When mRNA is produced, it has the one long hybrid protein transduction, hybrid ORF in a way. And then upon translation, it would produce a protein like this, which has OCT4 and also the protein transduction domain. So if we have a cell and we treat the cell with these kinds of proteins, the protein transduction domain may allow the entry of the OCT4, which is covalently linked to it, into the cell. And that is how the cell may have OCT4 in the cytoplasm. So this is the general idea, and people have been trying with it. There are advantages of it, and there are disadvantages too. The advantage is there is no genetic manipulation, right? So it is a most safe way of doing things, but it is very inefficient. Inefficient because we have a cell, and the cell should have high concentration of 4 different proteins, OKSM, all together at a time. And then how can we ensure that all these proteins are in high concentration within the cell? That is a difficult one. Nevertheless, people have used these things to get as a proof of principle it does work. And they are using protein transduction domains for several other purposes too. So here are some of the examples, and what has been carried by these protein transduction domains into the cell is some of the examples here. If we were talking about this, for example, is a proof of concept that we could allow the transfer or transduction of the OKSM proteins into the cytoplasm. And here are some references if you want to talk about it or read about it. There is another method that is also kind of safer. We are saying safer because it's not involving any genetic manipulation. The idea is, what if we can use some molecules, and the molecules differentiated cells to induce pluripotent stem cells, or maybe from somatic cells directly we give lineage reprogramming. That means directly from one cell type we want to get beta cells from pancreas, or maybe we want to get neurons directly. We are always talking about somatic cell giving rise to or making induced pluripotent stem cells and then deriving a cell type and so on, isn't it? What if we can do like this? That is somewhere it comes in transdifferentiation. That means one lineage of cell is being differentiated into another lineage of cell. So several labs have tried out different synthetic molecules, and there is a huge list of those, but I'm not going to discuss all of them. But what we can try to know is there are possibilities where there are several compounds. We don't need to mug up all these things, but they can act on several other enzymes or genes, say DNMT, de novo methyltransferase, deacetylases, and so on, which can either inhibit or activate the epigenetic remodeling or reverse the differentiation, because of which the cells might become induced pluripotent stem cells. So I will have some links here which you can try to look at, and then you probably will be able to know some of the examples. Here is a whole nice list or big list of those things. I don't want you to remember or mug up anything, but you should be able to uh, know at least two examples. Okay, you can choose whichever you want and then be able to answer them in the exam. So valproic acid, for example, is a is an inhibitor of histone deacetylases, and then the consequence also may be there. So the agenetic methods are what we have discussed. One of it is using protein transduction domains. We have also discussed about small molecules, synthetic molecules that can be used to directly for reprogramming to happen. There are other methods, the genetic methods, which some of them say you could have cloned OCT4, c-MYC, SOX2, KLF genes into retroviral or lentiviral vectors and then allow them to infect the fibroblasts or some other cell types. And then when the genes get overexpressed, then they become reprogrammed. So in retroviral and lentiviral vectors, what happens? If we clone them in 4 different of these like this, then the probability that one cell will be infected by all the 4 is very difficult. Maybe they would have done it with serially. Once first OCT4 is transfected, and then c-MYC, and then SOX2, and then KLF4. So we finally may have a cell which is infected by all the vectors, OKSM. So in lentiviral and retroviral, what the goal is that the genes get integrated into the genome and then they are overexpressed, because of which these OCT4 or other genes could be expressed and then pluripotency might have happened. The point is that it is employing genomic integration, because of which there is always a risk of tumors, and that is not a safe way at all. There are other viral vectors like adenoviruses. They enter into the nucleus and they remain integration. They do not integrate into the genome. They remain separately like this. And from there, they could express OCT4, for example. They are much better than the retroviral one, because there is no integration and because of which the viruses are not there. But then we never know what is happening to this genome at the end, or the viral vectors. Are they integrating or are they degraded? It's always a risk in that sense. Some people also use something like this, plasmid transfection, or without viruses, they would just make some episomes, and with 2 or 3 genes like this, one episome with 3 genes and another one with c-MYC, and then transfect. And upon overexpression, reprogramming happens. So ideally, what do we want in an induced pluripotent stem cells? As I was describing in the beginning, we do not want any kind of genetic changes to the genome. That is the first thing. We do not want any kind of risk in that. We should have appropriate expression of OKSM so that reprogramming can occur, and eventually we want to achieve that the reprogramming occurs. I'm going to show you one of the papers that I like about it. I'm going to give you only the essence of it. If you are interested, you can read more on that. Do you remember one of the goals is something like this? I have a genome, and having genetic manipulation is bad, but it is the only efficient way. Then how can we do it? What if I have all these 4 genes like this? Okay. And then I integrate them into the genome. After reprogramming, I delete them. I restore the way the genome was. Then we are making it efficient process, efficient reprogramming. And secondly, we are retaining the genome as such. No problems there. right? That is the idea some people got. And can you think of any ways how integration can happen, and then you can also delete any? Can you think of any genetic elements that you may know? Anybody? In the last course, we learned about transposons. Transposons are jumping genes. They move from one place to another place. There are two types, of course: cut paste and copy paste. For our purpose here, we are only discussing about cut paste. Cut paste means they would come, integrate into one place, and they would have sliced up again from here and move to a different place. That is a cut paste mechanism. They can be used for the strategy that I was discussing, where we could transiently integrate them and at a later time delete them, right? I also want you to recollect what would happen if this is the target site, how transposition usually occurs. It would make a cut here and here, something like this, and G, C, T, G, A, and then A, C, T, A, T. The, the target DNA becomes like that. Transposon goes in there, and then the gaps are filled up, right? In the original target, we had only one ACT sequence, but now we have two ACT sequences. That is called as TSD, target site duplication. When this transposon moves away out of the target, what is left behind? A footprint of the virus that it was at that location, of the transposon, like this: ACT, ACT. Is it the same genome? No, the genome is not same. So how can we use that? Use transposons. That's a difficult one. And in that sense, they started using something called as piggyback transposon. Piggyback transposons are special because they do not leave any footprint. Okay? They do not leave any footprint. Maybe in the next slides, I will try to see if there is time, I will make it happen. So what they have done in this particular vector is they cloned OCT4, followed by SOX2, followed by KLF4, followed by CMYC. Instead of giving 4 different viruses and infecting one target cell, they have taken all the genes together like this. And then they have used a CAG. This is a promoter and polyadenylation tail. I hope you remember that eukaryotic mRNAs have poly A tail on the 3' end. And then we have, these are for selection markers, that is purine or neomycin phosphotransferase marker, and thymidine case for, this is for positive selection and this is for negative selection. You don't have to go into these details. I usually take it in RDN in medicine in your 7th semester. If you're opting there, I discuss more of these things. So the goal is they produce one mRNA and they produce this piggyback transposon. It does not leave a footprint. All of this from here to here, all of this gets integrated into the genome. First thing is that. And integrated, these genomes also, the transposons have these terminal repeats, right? Inverted terminal repeats, which are important for transposition to occur. So we take, they have put in the transposon into the genome, and then all these will be expressed from a single promoter, because of which an mRNA is produced. And if you remember again, prokaryotic mRNAs are monocistronic. That means one mRNA has only one open reading frame. Mature mRNA has only one open reading frame, and it will produce only one protein. But here they have cloned all the genes together. and they actually made it as a translational fusion. That means here is the start codon, and at the end you'll have the stop codon. These are all continuous actually. This is the stop codon. So when a protein is produced, it will produce something like this, a, a single polypeptide which is OCT4, SOX2, KLF, and CMYC. And this is not the same as having OKSM, right? They are fused. They cannot be functional. So they have engineered self-cleaving peptides between them. That means upon translation, this peptide here will cut itself there like that, like this, and like this. So because of which we will get OK, OSKM individually, and they can perform the reprogramming. Okay, you don't have to really know all these things, but it will be nice if you can go to the paper and try to understand it. I think you have enough grounding to understand if you're interested in it. I'll just leave you with this. Why piggyback transposons do not leave any footprint? Piggyback transposons have, this is the transposon from here to here, and the transposase would recognize them like this somewhere, and it would make a cut here and here, and also here and here. The part, this TTA, is made into a hairpin loop like this on both the sides. Okay. And when it goes to a new target site, it has a preference for sites like this, TT, AA, AA, TT. It's not like transposons, like other transposons, which randomly integrate into any place. Once they are integrated, this hairpin loop here is cut again. Okay, so you will get something like this. This is perfectly base pairing with the target site. And so it integrates. The original target site had only one of TTAA. And upon transpogenesis, you had TTAA and again TTAA. That is target site duplication. Look at the same here. You have one target site. There are two duplications of the target site, right? That is target site duplication. When the transposon is moving from here to a different site, what is happening to the donor site? The donor site has AATT here, and here also AATT if you're reading it the reverse way. And these are perfectly base pairing, and they will just be base pairing. And what are you left with? The original one, which is TTAA. Here we had duplication because of transpogenesis, but upon excision and removal of the transposon, we are again retained back to TTAA. And that is why these scientists have used piggyback transposons. Okay. Yes, it is. It involves a lot of molecular biology, and I think you people are good enough with that. You can actually have a look at the paper. And the paper is called, I will send you the link if you want. Let's stop here. If you have any questions, I would be willing to take. Anybody? Any questions? Okay. If you don't have questions, then I would want to, one second. I had some questions that I want you to be aware of. So we will try to see if you can. Mm. 1 second. Be online, please. Okay, I want you to, some of the questions that you have to remember are something like this. What are the different covalent covalent modifications that are there in for modifications in DNA and histones? You should know the chemistry. And what are the molecular mechanisms of X chromosome inactivation? Then what is imprinting and what is its role in development of zygote to embryo? And describe the different reprogramming methods with specific examples. That is why in the PPT I have given you some, and I would want you to pick up 2 or 3 examples each. Okay? Yes, I'll stop here. Do you have any questions?
No, sir.
Okay. Then take care, and we'll meet again in the next